Hey grade 10s, welcome to the last little section of this work. After this we're going to be starting with a process called mitosis and the reason for mitosis, why it's important, why the body does it and how it does it. It would have been a lot easier to be able to draw it on the board for you, but this platform will have to do. So we'll finish this today and then we'll probably get to mitosis tomorrow. The mitosis I'm going to put up is probably going to be the equivalent of 2 lessons because sometimes it takes really long. But I'll let you know according to the amount of time it takes and so on and so forth. But I hope it's going well. Alrighty, so let's finish this off. These are the last couple of organelles, and the organelles I'm about to speak about, they all are in both animal and plant cells. Okay, so we've got the endoplasmic reticulum. It's a branching network of membranes that runs through the cytoplasm. I have included a diagram at the bottom over there just so that you can actually see what the endoplasmic reticulum looks like, and there's some extra words on top of it, and that is the rough endoplasmic reticulum and smooth endoplasmic reticulum. What you can also notice is that the rough endoplasmic reticulum is directly attached to the outer membrane of the nucleus, whereas the smooth endoplasmic reticulum is free floating. Okay. And I'll get to why they call them rough and smooth in a bit. So what do? They transport substances from one part of the cell to another. So in a test or exam, you'll just answer transport substances and I'll be happy. If they are rough endoplasmic reticulum, which are those the piece with the red dots on, it means that they have ribosomes attached. And if they are smooth, it means that they do not have any ribosomes attached. And if you remember, ribosomes are used for protein synthesis. They're often connected to the cell membrane or the nucleus, as I mentioned earlier. Okay, so what I would like you to do in this particular slide is I want you to draw the nucleus. I don't want the detail on the inside, okay? I just want the outer layer and maybe the nucleolus. Then I want you to draw some rough endoplasmic reticulum on the outside of that, and then draw a free floating smooth endoplasmic reticulum on the other side. Please make sure that you label rough endoplasmic reticulum and smooth endoplasmic reticulum, as well as the ribosomes. Okay. This is just a 3D version of exactly the same thing you saw earlier. Right at the top towards the right, you can see the word nucleus. So that is a nucleus directly over there, and you can see it is double membraned. And on the outer membrane, we're going to see, or the outer nuclear envelope, we see this rough endoplasmic reticulum. Now, if you remember, I briefly mentioned earlier this year that that DNA creates RNA, and that RNA makes protein. So this rough endoplasmic reticulum with the ribosomes on the top of it, they're going to be the site where a lot of that RNA is moved around inside so that the protein synthesis can actually take place. And then all the way on the far left hand side, you see your smooth endoplasmic reticulum. So let's talk about the little ribosomes. Where they're found, they can be found free floating or attached to the endoplasmic reticulum. It's important that you understand that ribosomes are pretty much all over the place. We often find that they are free floating, and they can actually be made up by 4 separate proteins, which is pretty cool. But they are often attached to the endoplasmic reticulum. We found ribosomes in the mitochondrial matrix. And then there's an electron micrograph or transmission electron micrograph of a rough endoplasmic reticulum on the right-hand side over there. You can see that it is quite folded, and it is extremely folded so that we can increase our surface area, which means we can produce more proteins, we can transport stuff quicker. Now, these ribosomes contain RNA and protein. They are involved in protein synthesis. So it's a bit ironic that the RNA and protein that makes up the ribosomes actually reads RNA to make protein. They're made out of 2 subunits, the large subunit and the small subunit. I don't expect you to be able to draw this. In a test or an exam, you would just draw a small little dot. I'm not really fazed about you drawing this. Alright, let's talk about the Golgi body. There's also other names for it. We can also call it the Golgi apparatus, and we can also call it the dictysome, the DITY SOME, the dictysome. All right, so those are the 3 words: Golgi body, Golgi apparatus, and dictysome. I will use those words very much interchangeably in a test or an exam. What are they used for? They secrete the secretion of useful substances. So what happens is the what do you call that thing? The nucleus creates the RNA. The protein is made in the endoplasmic reticulum. It is then moved from the endoplasmic reticulum to the Golgi body, where the proteins are folded and they're fixed up, and then from there it is pushed out the cell. Okay, so it goes baby step through the whole thing, and they're all very much interlinked as well. So what do they secrete? Hormones to different parts of the cell, or into the cell or out of the cell, and they're often found in your glands. Now, if you don't know where your glands are, like your salivary glands at the base of your mouth, the glands like your adrenal gland, your pituitary gland inside of your brain, your hypothalamus, your pancreas, a lot of them contain these structures so that we can actually produce a lot of these useful substances. So if we're talking about secretion of hormones, which is a protein, do you think we could also talk about the secretion of enzymes as well? Very much so. All right, here's a 3D version of what's happening, and it almost is like when you are holding up flashcards in front of kids, and you take the front one and you put it at the back. That is ultimately what's going on. So when the hormones or the protein substance is moved to the Golgi body itself, parts of it actually break off on the sides. And when it breaks off, and please remember this also looks like a Wi-Fi sign, when it breaks off, it becomes smaller, and then the next piece moves forward, and so on and so on. These pieces move from back to front and back to front and back to front and back to front, allowing for new substances to constantly move on. We'll talk about the vacuole now. It is extremely large in plants, and it is small or absent in animal cells. They are large liquid-filled cavity bound by selectively permeable membranes, and that membrane is called the tonoplast. You need to know that word. I like asking it. I was also really ugly once, and I asked students to draw the vacuole for their cycle test, and it didn't end up well. So we'll draw it in the next slide, but please remember that I could often ask this. And then the contents of the vacuole are called the cell sap. So I want you to pause over here, and I want you to draw that piece at the bottom. I've added in a more 3D version at the top over there just so you can have a look. So I have put a vacuole in there. You can see that the darker blue on the outside is called the tonoplast, and everything on the inside is called the cell sap. This tonoplast plus cell sap together make a central vacuole. It's not that the central vacuole is a separate label all by itself, okay? So just a reminder, the tonoplast is semi-permeable, which means it only allows certain substances in and out. And then the cell sap, and this is important, I like asking this as well. Cell sap consists of water, dissolved sugars, salts, and sometimes pigment. So let's quickly talk about why I want water in there. You remember when we did inorganic substances, that water was a good transport medium. It also allowed for a lot of chemical reactions to take place, as well as it is something that can dissolve a lot of substances. So I want to fill that entire structure up with water so that I can dissolve substances such as sugar. Because remember, sugar is going to move around where osmosis is going to take place. So if I have too much sugar in one area, it's going to cause a whole lot of water to follow with it, because it's just that it has less free water. Also includes salts. So, and sometimes to a large extent as well, in amoebas sometimes we can store dissolved waste products in there as well. So you can remember that, like when this actual vacuole itself can start growing on the inside, and eventually we're going to remind you about turgor pressure a little bit later, but it's because we move all those salts and sugars directly inside over there. And sometimes there's pigment, sometimes there's carotenoids, sometimes there's xanthophiles. It just moves out and actually gets inside over there. So I expect you to draw that diagram at the bottom over there with a nice big heading, diagram of a vacuole. All right, so let's look at some of the functions of the vacuole. We have a turgid cell and a flaccid cell. This has to do with turgor pressure. So the water enters by osmosis. What is osmosis again? The movement of water from a high concentration to a low concentration down a concentration gradient through a semi-permeable membrane until equilibrium is reached. Okay, so it enters by osmosis. The vacuole swells because all the water is moving inside of the vacuole. So it is endosmosis into the vacuole, and this pushes against the cell wall, causing the whole structure to become rigid. Whereas with a flaccid cell, the water lost from the cell, and that's because glucose and or salts have been moved to the outside of the vacuole, which causes it to now all the water must move out into adjacent cells or just into the cytoplasm. This causes the vacuole to shrink and the cell loses its shape. This is when we say a plant has wilted. So you haven't watered it for a while and now all of a sudden it's all droopy. In some freshwater animals, like the amoeba, a specialized vacuole is found, or a contractile vacuole. All right, we did this just the other day when we did osmosis. It actively pumps fresh water out of the unicellular organism, such as an amoeba. Then we have a food vacuole, also known as a phagosome or phagosome. Phago or phago means to eat. Okay, and we did briefly mention this as well. And then finally you have lysosomes. This is so important. Please make a big old NB next to this and highlight it. So lys means to split, and some is to do with a vacuole, okay, or a vesicle which holds these digestive enzymes. So it's a specialized vacuole that contains digestive enzymes. So now I want you to recognize that when this little amoeba is floating through water and it has eaten food through the process of phagocytosis, it needs to be able to break down that food. And the way that it does it is that the little vesicles containing digestive enzymes or lysosomes attach to that phagosome and actually spill all the digestive enzymes inside, and it rips apart all that food. Once it's ripped all that food apart, all the leftover enzymes, so on and so forth, are moved to the outside of the cell, much like a contractile vacuole, and the contents are spilt out so that it doesn't mess up with the osmotic balance. Almost finished. So we got the cellular cell wall, rigid outer covering of only plant cells. Only. I'll teach you next year that bacteria also have a type of cell wall, but it's not the same as plants. This is cellulose, okay? And remember, anything ending in an ose is a carbohydrate. So cellulose is a carbohydrate that makes up the cell wall. It is non-living, therefore not part of the protoplasm. Remember, the protoplasm is only the living components of the cell. The first layer of the cell wall secreted by the protoplasm is called the middle lamella, and you can see it as the lighter green layer of the picture in the background. And then we have the second layer, which is made up of cellulose, and this is called the primary cell wall, and that is the more neon green on the far outside of the entire structure over here. The functions of the cell wall: it's to enclose the plant cell, forms an outer protective layer, gives rigid support to the plant and the cell. It is completely permeable and allows most dissolved substances through. So if it is small enough to get through the pores of the cell wall, it will get through, okay? So permeable means that anything can move through it as long as it is small enough. It doesn't choose, which is like semi-permeable, which means it selects what it wants to go through it, and non-permeable, meaning that it doesn't allow anything through it all. Then we finally have the middle lamella, and this binds adjacent cells together, and we'll talk about that a little bit more when we do mitosis of plants. All right, now please draw up a comparison table of only the differences between plant and animal cells. Okay, so here we have lysosomes in plants, animals, chloroplasts in plants. Remember, you can also add in there chromoplasts as well as leucoplasts. Cell wall in plants with a cell membrane. On the other side, you can say just cell membrane. In animals, many small vacuoles, small to no, and whereas in plants it's one large. Animal nucleus usually in the center, whereas in a plant, and that's because of just where we want the center to be, where we want our brain to be, whereas in plants, it's pushed to the side because of that large central vacuole. Finally, animals, their shape is rounded or amorphous. They don't have a particular shape, whereas in plants, they usually have a rectangular-ish shape. All right, so I want you to draw up this table, and that's the end of this little chunk. Well done, grade 10s. I'm extremely proud of you. We've been through a lot of work, and I know I spoke quite fast through this entire little section, but please take down some notes, and if you have any questions, don't worry to ask. All right, have a good day, and we'll start mitosis soon. Cheers.